Welcome to this ReadStore tutorial where we want to discuss the ReadStore ProData feature. The ProData feature enables you to store process data from your bioinformatics processing of raw data sets in the ReadStore database, and this is very useful, for instance, if you want to use ReadStore as input for your downstream analysis scripts that usually use some kind of process data for those analysis. One example is, for instance, single-cell RNA sequencing, where we often integrate gene expression matrices that come from different experimental conditions or replicates. And in this example, we have 3 replicates each for 2 conditions which we would want to integrate into one analysis. We can now pull all this information along with the experimental metadata from the ReadStore database, which saves us the effort of searching for those files in our file system and then looking for the metadata and a different location. And that in the end helps us to standardize our processes and make our analysis a bit more efficient. So here's now a more formal overview of how the ProData feature is integrated into the ReadStore workflows. As usual, we would first upload the experimental metadata and the raw sequencing files, for instance FASTQ files, into our ReadStore database, where those files are organized as datasets. And those datasets we can then access and integrate into our NGS processing bioinformatic pipelines. So these pipelines are, for instance, coming from your kit vendors to process the raw data into the respective outputs. Other examples are custom scripts that you build for alignment and quantification, or for instance, next flow workflows. The output of these processing steps are then the pro datasets, the process datasets that of course depend on the type of assay that you're running. Some examples of these are, for instance, single cell count matrices that come from single cell omics experiments, or variant files for genomics analysis. And these process data sets can then again automatically be uploaded again and attached to the data sets in your ReadStore database. With this, you can keep track of all your raw and process data along with the experimental metadata at a single location. And for your downstream analysis, you can then, of course, go back to your ReadStore database using the Python or R integrations or the command line interface and retrieve access to those process data sets without having to search for the data sets on your file system. So let's go into the command line and see how we can upload process data, pro data sets, and attach them to individual data sets in our ReadStore database. For this, we're going to use the ReadStore command line interface. And it's important in the beginning that your user account has staging permissions if you want to upload or later delete process data. If you need more information on the staging permissions, I'm going to link this here, the video where I explain more about this topic. And now, before we go into the individual commands, there are basically 4 operations that you can execute. The upload, list, get, and delete operations. And we are now going to go through each of them individually to see how we can manipulate those process data on our database. So let's start with the ReadStore upload command and check what we need in order to upload a process data set. For the upload command, there are a number of arguments that we need to set. So we have to set either the dataset ID that we want to attach the process dataset to, or the dataset name. We need to give the process dataset a name to access this later, and a type. So a type is just a string value that describes what type of process dataset this is. So this helps you later to also filter the process datasets based on their content. So for instance, this could be gene counts for RNA-Seq or bat files or any different type of process dataset. You can set a description here as a string, and you can also define for the process datasets metadata keys. So these are again a JSON formatted string with a key value pair that you can later query. So we prepared here one file that we want to attach now to a data set in our ReadStore database. This one is a single cell count matrix for a mouse control replicate 1, and we want to attach this now to a data set in our ReadStore database. And we see that we have a mouse control rep1 dataset, and this is the one that we want to attach our gene count matrix to now. For this, we run the upload command. So here I prepared the upload command, and the upload command consists of different arguments. As I said in the beginning, we're going to attach the gene count matrix here to this mouse control rep1 dataset, which we assign here with the -d attribute. Then we give this process dataset a name in the database, which is mouse control replicate 1 DGE. And with the -t, we can set a data type. So we can define data types by ourselves. There are restrictions, and here we call this gene count matrix. So if we upload also gene count matrices for other data sets, we could later then group them by this data type. And the last argument here is the path to the count matrix file that we want to upload. And if we execute this, we see that we get a message that we successfully uploaded the dataset. So let's check if our dataset has been registered in our database. For this, we can use the ProData list operation. And here we see that we have our Pro dataset with the ID 12 and the name that we supplied. Gene count matrix is the data type, and then we have the version. The version increases if we upload a file with the same name for the same dataset again. So this is very helpful if you have pipelines that you need to update, for instance. Then you can automatically archive an older version of this process dataset. And here we have the dataset ID and the dataset name that the pro data are associated with, as well as the path. Now, what we want to do is use the get operation. The get operation always returns a single dataset with a bit more information than the list. So here we see that we can retrieve pro data by their ID or by a combination of the dataset ID or the dataset name plus the name of the pro dataset. And we have options to return only the metadata if we've set them, or the path to the file, which is useful if we want to integrate this command into an automated pipeline. And we can also select certain versions of the file in case we want to go back to an archived version of this process dataset. So, and the idea of our upload dataset was 12. And here we see that we are retrieving information on this process dataset. There's quite a bit of information here, but those are now the attributes of our process dataset 12. Including information on where the file is stored and metadata and a time when it was created. Last, we want to look at the delete operation. Here's an overview of the parameters that are required to delete a process dataset. It's important to note that the delete operation is only happening on the database level. So it's not deleting the actual file on your file server. They're still there and protected. It's just removing the entries from the database. And here we see that we need to provide either a ID for the process dataset that we want to delete, or a combination of the dataset name or the dataset ID that the process dataset is attached to, plus the name of the process data, and also optionally a version. So let's prepare the command. So if we look at this command, this now deletes the process dataset mouse control replicate 1 DGE that is attached to dataset mouse control replicate 1. And if we execute this, we get a message that the process dataset with this ID has been deleted. You can find more information on all of these functions in the manual of the ReadStore command line interface. And so finally, let's look at the ReadStore app and where we can find the process data in our app. And for this, we move to the datasets page here, where we find an overview of all our datasets. And we saw before that we uploaded a process dataset for our mouse control replicate 1 dataset here. So we can just select this and then move down. If we have the detail view activated, we should see the different tabs here. And in the ProData tab, we should get an overview of all the process data sets that we uploaded for this data set. And with this toggle, we can also include archive data sets. So we see here that there has been a first version of this gene count matrix, which we have overwritten then with our second version. If we select individual process data sets, we can get access to the detail view. And here we get a summary of the data set plus access to the description and metadata and the file path. And we can also delete those process data sets from the user interface here. For this, we go to the update section, and in the ProData tab, we have the option again to select the data set and delete it. This has been the overview of the process dataset features that ReadStore offers to store not only the raw data, but keep them connected to process datasets that you can use in your downstream analysis. If you have questions or comments, please leave them here. If you want to use ReadStore, you can try the demo version that is available. I'll leave a comment in the notes here. And otherwise, I wish you a great day.